From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode is recorded on April 25th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 89th episode of Matters Microbial. I'm truly grateful for every download, listen, and view on YouTube. Thank you, as always, for spreading the good microbial word far and wide. I'm just delighted that my fall microbiology course, the last time I will probably teach at the University of Puget Sound, is completely full with a larger enrollment than normal. They'll be my last crop of in-class micronauts. But don't worry, I'll still find a way to promote microbial centrism, I promise. Today's topic is very important to me, and it should be to each of you. Most of us have heard about the growing threat of antibiotic-resistant disease-causing microbes. Bacteria are ever mutable and exchange genes with ease. So many antibiotics that at one time were used to treat bacterial diseases are no longer effective. This is a constant issue in the treatment of bacterial disease. This image from a CDC report, posted in the show notes, states that more than 1.2 million people worldwide are killed by antibiotic-resistant microbes each year. This was from 2019. In the United States, almost 3 million antimicrobial-resistant infections occur each year, leading to more than 35,000 deaths. And the estimated costs of dealing with this growing crisis in the United States is at least $4.6 billion per year. And this is why I believe that each and every introductory biology class should cover this topic for at least a week, probably two. It's so very relevant to everyday life. What do I mean by this? What do I teach my students about it? Here you can see the chemical structure of one common antibiotic, ampicillin. Please note the squarish structure in the middle of the molecule. But how does ampicillin impact bacteria? As you can see here, ampicillin prevents cross-linking in the cell wall of the bacterium. The bacterium is thus very fragile and dies easily. Since the hand of Darwin is on all that lives, it should not surprise you that bacteria have evolved genes to deal with this and related antibiotics. One such gene encodes the enzyme beta-lactamase that cuts ampicillin and related molecules in that squarish region, making the drug ineffective. Here's an experiment I do with my students every year. Imagine a plasmid carrying that ampicillin resistance gene and a gene for green fluorescence protein. On a petri dish that contains the drug ampicillin and many sensitive bacteria, the bacteria with the plasmid grow well into colonies, as you can see, and they glow under ultraviolet light. But after a few days of incubation, you can see the green glowing colonies are surrounded by non-glowing satellite colonies. Now, if you pick a glowing colony, A, and a non-glowing colony, B, you will learn that only the glowing colony can grow on ampicillin media when restreaked. Remember, the glow means the plasmid with ampicillin resistance is present. Why? The beta-lactamase enzyme is secreted by the resistant bacteria. The area around that colony, that glowing colony, is depleted in ampicillin. So the sensitive bacteria surrounding those green colonies become able to grow because the antibiotic is being destroyed over time. I can even get creative with the process, as seen by this movie. Friends, this is just the very tip of the antimicrobial iceberg, but it is a way to remind students right in front of their faces of the antimicrobial resistance threat that we all face. So, I'm beyond honored to welcome Dr. Amir Mitchell of the University of Massachusetts Medical School to the podcast to discuss how artificial intelligence might be used to keep us ahead of the ever-shifting threat of AMR. Welcome to the Quality Quorum, Amir. I'm so happy to meet you.
Thank you very much, Mark. It's a pleasure to be here.
I have so enjoyed looking at some of your publications and your wonderful website. I especially appreciate your outreach work for obvious reasons.
Thank you very much.
I think that the first thing that we ought to talk about, if you don't mind, is a little bit about what motivates your research and just take us step by step through it.
Right. So, to be honest, my lab is interested in a very wide range of topics. If I have to sort of describe it all in a single image, I think you can pull off slide number 2. So I'll jump ahead. And that shows the main question my lab is interested in, which essentially is this triangular interaction of the interactions between bacteria, the microbiome, the host, and the different drugs. We ask various questions about these triangular interactions, like how bacteria adapt to different niches in the host, whether it's the gut or even completely new niches like solid cancer tumors that we know are infected sometimes and get colonized by bacteria. We ask questions about how bacteria interact with one another. And many of the questions we ask are also about the interactions between specific drugs and the bacteria. So these are, again, in very broad strokes, our regions of interest. And I would say this is probably just some of the questions we're asking about this triangular interaction. I find many, many different corners in science interesting. And if you take a peek at my publication record, I've asked many, many different questions in very different model systems across the years.
You know, for listeners and viewers, this topic is more relevant than you know. One of the things that always confused me is how certain drugs appear to work quite well on some individuals and not others. And in some cases, it has to do, as you allude, to the microbiome being different between those individuals. I know this is the case for certain chemotherapeutic agents. And so understanding that relationship is so important in ways that someone might not expect.
You're absolutely right. The microbiome seems to be a major participant, a major factor that dictates how we respond to different diets, to different drugs. Also for drugs that are completely what we call non-antibiotics. Mm-hmm. These are drugs that we never use to treat infectious diseases, yet the microbiome seems to play a major role in how effective are these drug treatments. And that was one of the major observations that lured us into this initial field of drug microbiome interactions, trying to understand why some bacteria respond to drugs that were never designed to be being used as antibiotics. And I think it was about 5 years ago that we read this publication by the TPAS lab at EMBO, at EMBL, putting a number to this phenomena. They did a very, very extensive screen, and what they suggested there, what they observed there is about 25% of all of these drugs, these non-antibiotic drugs, again, drugs like chemotherapies, psychiatric drugs, diabetes drugs, they're never used as anti-infective agents, yet a quarter of them actually target bacteria, either pathogens or microbiome or species in our microbiome. So since that initial observation, we were kind of really, really fascinated by the question of how are they inhibiting the microbiome? Why is it that these molecules that were never designed to be antibiotics actually targeting the microbiome? And I think this is extremely interesting because either answer to that would be interesting. If they operate just like antibiotics, then there's a real risk that if we take these drugs chronically over years and years, sometimes we take these over decades, we would unintentionally develop antibiotic resistance, within our microbiome. So that's one reason why this is important. The other, the sort of flip side of this, if these drugs kill bacteria in completely new mechanisms or completely new ways, what we call mechanisms of action, then maybe we have promising leads for the development of future antibiotics. So I think for us, it was really, why are they killing? How exactly are they killing bacteria? And we realized that no matter what we do, we'll get something interesting out of it.
That's the best kind of research to have.
Yeah, absolutely.
You know, there's something I tell my students about, and it's not directly related, but it's involved. Wood rats are able to eat creosote bushes, and creosote is this like toxic polyphenol. Mm-hmm. And their microbiome contains participants that are able to break that down and detoxify it. If you try to feed regular rats creosote, they won't eat it. And if you force it into them, they will become hurt or damaged. So in one case, we take those wood rats and we cure them of their microbiome through antibiotic treatment. And then they find that that same creosote that they've been eating a very long time is toxic to them. And conversely, a microbiome transplant into a laboratory rat gives them that ability. And this emphasizes the role in a natural world that the microbiome can have in unexpected ways.
Absolutely. We see this also sometimes in drug therapy. The microbiome can sometimes inactivate drugs, degrade drugs. The microbiome can activate some drugs. That can happen in the gut, in the digestive tract, but it seems to maybe also happening in infected tumors, tumors that host bacteria that can potentially degrade frontline chemotherapies, and by that might protect the tumor niche from this chemotherapy. Now, this is still under question whether this really happens in human patients, but it is intriguing to explore these questions, whether the microbiome that the host has, either in the digestive tract, on the skin, or in infected tumors, actually interacts with these frontline chemotherapies, and by that might protect or might even sensitize the tumor to the chemotherapy. And so some of our initial work actually started to look into that using various model systems like C. elegans, its bacterial diet, and different chemotherapies, or what we call cancer spheroids. So these are globular aggregates of cancer cells with a microbiome or infected bacteria infecting them and treating these spheroids with different chemotherapies and seeing how the bacterial genome, the different mutations in bacteria, might impact drug degradation and therefore chemoresistance and chemosensitivity. And the notion we were trying to sort of explore there, and again, I have to emphasize, this is really just an exploration at this stage, is whether this concept of gradual increased resistance against chemotherapy can come not from mutations in the cancer cells, but actually through mutations in the microbiome, either in the gut microbiome or in the tumor microbiome. So these were the initial sort of questions that brought us in or lured us into this fascinating topic of interactions between bacteria and non-antibiotic drugs.
It is really an interesting concept. I remember reading years ago that inside a tumor, it's fairly anoxic. And so you tend to concentrate particular anaerobic bacteria in there. And I knew some bacteria were kind of, they were swimming into the tumor, so they're attracted to it in some fashion. But now we're learning more about all of this and this relationship. So we really are a series of ecological niches, all of us.
Yes, absolutely. We are just floating niches that bacteria take advantage of. That's how I see us. And I have to say, we won't have time probably to go into that, but we are exploring this process of tumor infection and colonization. We are trying to understand what makes bacteria sort of find their way into the tumor, this homing into the tumor, and this very successful colonization of bacteria of the tumor niche, which we thought is very inhospitable, but bacteria seem to love, right? It's anaerobic. There's necrotic tissue there. There's lots of spillage of different nutrients there. Bacteria actually tend to like some of these areas in the tumor environment, and they can really thrive there. So we have some work now that is still in pre-publication, but that tries to really look into that in a very sort of high degree of detail, the population dynamics of bacteria infecting the tumor, which I think is fascinating, and we know very little of. We know we can see bacteria inside tumors. We know nothing about the gradual infection and colonization process. And I find these sort of niches of biology where practically nothing is known to be really fascinating. Right? This is like, this is the frontier. This is where you make discovery. And I just like being in that position with my students where we are the first people to ever observe a specific phenomena for the first time. So it's super exciting to operate there.
And it's important for listeners and readers to remember, and I've said this several times in the podcast, bacteria will evolve under laboratory conditions. I do it in my lab with undergraduates. Right. So is it a surprise that there might be a series of specific mutations that can enable some bacteria to do this colonization event? It's just yet another environment to be colonized, right?
Absolutely. I share that world of view with you. Bacteria are remarkable at adapting to new environments. There's no reason to believe they will not adapt to the tumor niche. Yep. And there's no reason to adapt to think that they won't adapt to chemotherapies that are very broadly cytotoxic agent. They poison the tumor. They also poison any inhabitant of the tumor. I share that worldview with you. They adapt very, very quickly. It's bound to happen. The real question is what exactly will happen? What will transpire through an evolutionary process? Absolutely.
I think that's a wonderful topic. And there I got overly excited, as I often do with cool work. That's just who I am, and I've always been that way. So perhaps we should move to the topic at hand?
Sure. Let's do that. So I'd like to first maybe talk about very broadly about antibiotics. So we can pull up slide number 3. Slide number 3 shows us a diagram that we made of the bacterial cell, this very generic bacterial cell, and the different antibiotics or the antibiotic classes that we know of. There are multiple antibiotic classes that have been developed mostly in the 20th century, in the middle or to the mid 20th century, and they attacked, and here you can see them in different colors, different fundamental processes in the physiology of a bacterial cell. They can, target DNA replication, or they can attack translation or transcription. We know a lot about these antibiotic classes. We know how they work. What we don't really know is how all these non-antibiotic drugs, and again, I call them generally non-antibiotic, they are antimicrobials. They were never designed to be antimicrobials. How exactly they operate relative to the standard antibiotic classes. So this is the larger framework in which we ask these questions, is how do these non-antibiotic drugs compare to the standard antibiotics? Do they operate in the same way, or do they operate very, very differently? Now recall that we are trying to answer this question not for a single drug, right? We're trying to get answers for dozens, if not hundreds of non-antibiotic drugs. So that is a really tough question, and you need to come up with a methodology, with an approach that will allow you to answer not for a single drug, but for dozens of drugs, what we call a high throughput assay, right? Something that will allow you to resolve or get an answer for dozens and dozens of drugs. So that's how we started off this research that I want to sort of expand on. By this initial observation made by another lab, dozens of these non-antibiotics kill bacteria. How exactly they do that? We know nothing. We do know a lot about standard antibiotics, and we want to make that comparison. So that was sort of how we started this approaching this question is by this real necessity of how do we even answer that, but not in the context of a single drug, but in the context of dozens of drugs. And that sort of led us down this rabbit hole, which I'll describe. So I think we can talk a bit about the methodology. And again, I urge you, Mark, to stop me if I get overly technical, but I do want to convey the basic concepts behind functional genomics, or these genetic screens, as we call them. So we can switch to slide number 4, that sort of gives you a rough sort of pipeline of how we do this work. So our work is leveraging on a method that has been developed for the past couple of decades at least. And that is the method of using loss-of-function genetic screens to understand a mechanism of action. So imagine this. We want to understand how a specific drug kills E. coli, our model organism. Okay? We know a lot about the biology of this model organism. We know it has about 4,000 genes. We know that just a few hundreds of them are essential genes. It cannot do without them. We know for the vast majority of the genes what exactly they encode for, what type of enzymes or functions they perform. And we'll use all that information to understand the drug mechanism of action. And the process sounds a bit complicated, but the principle is actually quite straightforward. The idea there is to start with a collection of different mutants. We have more than 3,000 different mutant strains. Each one of them is that E. coli strain, our lab E. coli strain. And each one of the mutant has a single gene that is knocked out. Now recall, we have some essential genes. We cannot knock these out, okay? We can only knock out non-essential genes. Genes that are nice to have, but are not necessarily critical for the bacterial survival in the standard conditions where we grow our bacteria. So we have that array of about 3,500 different knockouts. Each one of them is a single gene knockout. We take that collection of mutants and we do an experiment where we split it in two and we grow half of our cells without a drug and half of our cells with the drug. So you can see on the left-hand side this splitting into two growth conditions, with and without drug. And what we do is we use various sequencing approaches to try to quantify which mutants grew well without the drug and which mutants grew well with the drug, or which mutants were very sensitive to the drug. Mm-hmm. That comparison allows us to actually calculate what we call a fitness coefficient, how sensitive or how resistant each one of these knockouts or each one of these mutants is to the drug that we are studying. That actually tells you a lot about the drug mechanism of action. So just to give you an example, maybe before you ask your question, sure. Imagine a gene that encodes for a protein that helps with DNA repair. We know that if we take a drug that targets DNA replication, we'll have lots of errors in the DNA replication, and you will really need those enzymes that allow you to repair DNA damage. If you have a strain that is knocked out, that doesn't have that function of DNA repair, it will be especially sensitive to that type of antibiotic. An antibiotic, fluoroquinolones, that target DNA synthesis and replication.
For listeners and viewers, this is so fantastic because the advantage of working with E. coli is there's so much known of it. And I've used what's called the CHIO collection, which are these single gene knockouts. And they're wonderful because if you want to know what the phenotype is, if you knock out a specific gene, you can do that. There are a lot of bacteria you cannot do that for. And I want to remind everyone, once we understand how E. coli does something, we can often expand that to other related organisms that might be of medical relevance. So it's a really, really powerful technique. And what I like about it is the people who developed that distribute it.
Absolutely.
And so other people can study these things. Amir, just so you know, I mean, that was something that was done many years ago. We were trying to find what genes in E. coli might help protect them against the predator Delovibrio. Mm-hmm. And we used a Caio collection to try and look into that. We found out that we couldn't find anything that was important, but it's still the right approach.
So I'll just mention, you mentioned the Caio collection. It's a tool that was sort of cloned and put forth as an open resource about 20 years ago. And the same guy, Hirotida Mori, was generous enough to share with us this new collection of knockouts. It's almost identical to the Caio collection that you mentioned, but it has a beautiful additional complication. Each one of these knockouts has a barcode, a unique 20 base pair nucleotide sequence that allows you to identify that specific knockout by pooled amplicon sequencing. That allows us to take the same approach people have been using for a couple of decades now, doing these genetic screens, but to really push the envelope on the throughput, on how many experiments we can do, right? So that, and again, serendipity. We met Hirotida Mori randomly at a conference. He was showing a poster about this new resource. We were super excited. We asked him. He was super helpful and willing to share it. And he shared with us something pre-publication. So our first publication was with him using this resource for the first time. And that really allowed us to ask the question. And it was purely based on the generosity of other scientists we randomly met in a conference.
This is one thing that I really get concerned about. Sometimes scientists are portrayed in the media, gently put, quite differently than how they are. And I'm not saying that there aren't people who are egotistical. I'm not saying that there aren't people who are very unpleasant. Of course there are. But I have found in the microbial sciences, it's a culture of sharing ideas and information for the most part. And this is a wonderful story you just told. Listeners and viewers, you should know that because of the barcoding that you heard Dr. Mitchell mention, you can look at many of those strains at once instead of doing them one at a time. And so that really makes it, rather than 3,500 experiments, you can do it in just a few.
Absolutely. You're right. And we'll get to that soon, but I think that allows us, that was a technology that was developed by somebody else, but it allowed us to ask these questions in a completely different throughput than people did before. So I'll go back to the slide 4 that talks about this method, right? So we can take this collection and infer the sensitivity and resistant profile of this collection to individual drugs. And if you look at the center, we can repeat that for different drugs, right? So we have different drugs, and now we can do that experiment again and again and again, dozens of times, and get these sensitivity and resistant profiles of our collection under each one of these drugs. Now we can take this data, and I want you to think about these profiles as fingerprints. These are fingerprints that are indicative of how these drugs kill the bacteria. What are the processes they target in order to harm bacterial growth? And if you have two drugs that work identically or in a very similar fashion, they will likely have very similar fingerprints. Now, if you collect enough of these fingerprints, what you can do is you can start to analyze this data and look for drug-drug similarity. You can see how similar these two fingerprints are and convert this data, this huge matrix that we constructed of all the sensitivity and resistant information, into what we call the drug-drug similarity networks. That's what you see on the right-hand side of the slide. We can, for each drug, sort of represent it as a node in a network, and we can draw an arrow between two nodes if they are highly correlated or highly similar in their sensitivity or in these fingerprints. That allows us to convert all this data into drug-drug similarity network that you can see on the right-hand side. And when you see it in this way, it really allows you to sort of embed unknown drugs into this network, right? This is what we call the guilt by association. So I'll get to it in a second, this idea of how we add new drugs to this network. But for now, let's just say we have a very powerful method that uses information from this mutant collection to understand which drugs have a similar way of killing bacteria. And we represent that data as drug-drug similarity network, where drugs that have a similar mechanism of action, a similar way of killing bacteria, they show up as a cluster, as multiple points there that share all these connections between them, making them what we call tight network hubs. So that's the method, or that was the idea at least, that we thought could help us answer those questions about the mechanism of drug action for dozens of different new drugs. So we have that. We first need to see that it works, right? We can't just hand wave. But if this works, if this method works, then we can take a new drug, add it to that network, and see where exactly it lies. So let's think about 3 different alternatives. So we'll switch to slide number 5. You can think of 3 different outcomes from these networks. We have the networks. We construct them for all the classical antibiotics, for which we know what is the killing mechanism. And we'll get this cluster sort of diagram of the network. And now we're taking a new drug, it's marked here in black, a non-antibiotic drug. It kills bacteria, but we don't exactly know how. If we add that drug into our network, and again, we don't do it manually, a computer does it for us based on the sensitivity fingerprint, we can get 3 alternative outcomes. This mapping can tell us that for one drug, that it has what we call a standard mechanism of action. The star on the right-hand side of the slide is an example, it's a theory for now, of a drug that operates very similar to gyrase inhibitors, a very well-established class of antibiotics. That means that this drug, we don't use it to treat infective diseases, but it kills bacteria just like the gyrase inhibitors do. So it's what we call a standard mechanism of action. You can think of a situation where what we see is a drug that sort of moonlights and does 2 different things. That's the middle panel. Maybe this drug, this black diamond, works a bit similar to those antibiotics that target the ribosome, that target translation, but also a bit like those that target the cell wall. That drug might operate as a drug with a dual mechanism of action. It kills bacteria in 2 different ways. These are highly sought after drugs. And the last option, which for me is the most exciting one, is that maybe this drug operates in a completely different manner. It doesn't connect to any one of these standard antibiotic classes, meaning it kills bacteria, but it kills it in a very creative and new way, and therefore it's a potential lead for the future development of antibiotics. So these are the potential outcomes, and that's what we call the guilt by association. We try to associate our new drug with previous drugs for which we know what is the killing mechanism.
You know what I really appreciate about this approach is that many times, and I know this isn't directly what you're doing, we just look at, say, sequence similarity between things. The problem with that approach, and it's a very productive approach, is that you don't find things that are unrelated. So you entered into the experiment with an expectation. What I like about what you're doing is you have something where you've established this, it looks like graph theory to me, this network.
Absolutely, yes.
and then my wife's a mathematician. You have to forgive me for that.
Yeah.
But what happens is that now you have this new one you don't know, and you can plug that in and see where it fits rather than take something that's already known and modifying it, which is what a lot of antibiotics actually are.
Absolutely.
So no, this is exactly the approach that gives me hope.
Okay. So this was the theory. This was our ambition starting off on this, I would say, a very, very ambitious project that we didn't really know whether, would work. So think of it as a sort of a 2 or 3 tier sort of approach. We start off by just trying to build these networks for the established antibiotic classes. Then we'll do the experiments with these new non-antibiotic drugs, and we'll embed them into these networks. And the last thing is, for those that maybe operate in a new way, we'll try to use the information from the mutant library to understand, well, how exactly are they killing the bacteria, right? We have some molecular detail or some hints on how they kill bacteria. It won't remain a mystery. We have some clues on the killing mechanism. Okay, so that, again, taking us back 5 years ago, was our sort of, back of the envelope sort of plan. This is how we're going to take over this very ambitious project. Let's see if it works. So I do want to sort of, again, give the listeners and the viewer the sense of the scale of the screening we're doing or the data we're collecting. Myself, by training, I come from both a biology background and computer science background. And a lot of the things we've done, or I think some of the success here, has to do with the ability to use an interdisciplinary approach to sort of track this huge data set. And I want to give the listeners the sort of the sense of the scale of the problem we're dealing with, okay? And I'll talk about some numbers. So we can switch to slide number 6. And again, this is just to give you a scale of the problem. What we experimented was with a panel of almost 200 drugs, okay? Each one of them we assay independently. We have our knockout library that has more than 3,000 different knockouts. We have 2 of each. I won't get into that. We're doing the experiments in replicates because your listeners probably know that in biology, we always do experiments in replicates to have a high degree of certainty in our observations. So we do this in triplicates. And if you multiply these numbers, you see that we collect a heap of data, almost 2 million measurements, what we call fitness measurements, where each one of them tells you how well does a single knockout grow in the presence of a single drug, right? It's the fitness of a knockout in the presence of a single drug. It's a huge data set, right? There's no way you can open an Excel file and sort of find your way through all these numbers without applying some computational biology. And of course, my lab, one of the things it does, it combines both experimental biology and computational biology. So if I can give one piece of advice to the listeners, no matter where you are, who you are, at what career stage, learn how to program. Being a programmer is, your secret sauce. It's your superpower in biology research. And I think for us, it was extremely useful. Maybe we'll get to it in the end. I think the whole community is moving towards AI and, big data. I think it's a necessity. For the time being, if you're a person that can look at your results and crunch the numbers using computer programming, you have a superpower. And it's never too late to gain that superpower. So that's sort of my tangent that I always go through. This is a superpower. Biology is a big data science, and you have to have that superpower to succeed, I think, in that field. So we have that heap of data, 2 million interactions. We need to sift through that and to get those drug-drug similarity networks. And again, we don't have the time, but the process of going from the raw data all the way to these drug-drug similarity networks, these graphs, is, I think, a fascinating one, but not a very complicated one. Once you're a programmer and you look under the hood and we publish all the code, you'll see, you can probably write it today using ChatGPT within a few hours, right? There's nothing to it. You just need to understand the principles. Okay, So we understand the scale of the problem and what we try to do. Let's see if it even works, right? So I showed you these graphs of these networks and what we expected them to look like. Let's see if it really works for standard antibiotics. So we can switch to the next slide. That's slide number 7. And again, I'm jumping just to the meaty part of our research, not to bore you too much with the data crunching, but this is the drug-drug similarity network we actually infer from the data of our experiments, from these 2 million measurements of drug knockout interactions or drug mutant interactions. And what you can see, and for us, I have to say, seeing this image for the first time, I can still sort of recall back to that point in time with my student, Mariana Noto, who was the driving force of this work, and we'll get to her later. You'll hear about her in the future, I'm sure. Seeing this for the first time was a jaw-dropping moment for us because it told us, you know what? Our pipe dream actually works. You can take that huge data set and you can crunch the numbers, and it all falls down into these beautiful networks in which antibiotics that target the cell in the same way actually cluster together in this drug-drug similarity network. So let me just describe briefly what we're seeing. Here you can see the drug-drug similarity network. Each one of our antibiotics, and there's almost 100 antibiotics here, is a single node in this network. And we connect 2 nodes if they have a similar impact on our mutant collection. Right? Now, the algorithm knows nothing about what the antibiotics are. It only knows drug X impacted these mutants, drug Y impacted these mutants. Mm-hmm. To see whether this mapping, this clustering makes sense, we add the colors only after the computer finished clustering the similar antibiotics one next to each other. And it's just a proof of concept showing you that the algorithm knows nothing about how these antibiotics work, yet the signal we see in the sensitivity and the resistance is so strong that antibiotics that actually work similarly are bound together in these clusters, in these network hubs. And they're tightly sort of compacted together. Everything that targets the cell wall, all these beta-lactams that you introduced in the beginning, actually cluster together very tightly. And these are this yellow cluster that you see here. Mm-hmm. Everything that clusters, the folate biosynthesis cluster together. This is this green cluster here, right? So it works very, very nicely. But you can see something interesting beyond that. You can see some connections between the clusters. For example, antibiotics that are yellow, targeting the cell wall, have some connections to this red cluster that are antibiotics that target the gyres, that target DNA synthesis, right, and DNA replication. They have some shared toxicity mechanism between them. We know the details. It's really reassuring to see this. So for me and for Mariana, seeing this for the first time being plotted was sort of a jaw-dropping moment. Aha, it works. And it was super exciting.
That's quite a story.
So the method works. What about all these non-antibiotic drugs, right? Where do they fit into these clustering networks, all these sort of drug-drug similarity networks? And that was the next stage. So we took those drugs and we added them to the network. And again, the computer knows nothing about them beside their sensitivity and resistant profiles. And what we see is the picture that emerges in slide number 8. So if you can pull up that slide, what you'll see here again is a drug-drug similarity network. We still have the antibiotic clusters, and we add to the mix all these black drugs. These are the non-antibiotic drugs. Just to remind you, these are drugs that still kill bacteria, but we never use them to treat infective diseases. They kill bacteria for unknown mechanisms or unknown reasons. And again, what you can see here is quite striking. as you can see, most of these black dots that represent non-antibiotic drugs stay completely disconnected from the antibiotic classes. None of them is embedded within a class, meaning that none of these drugs is just an antibiotic or non-antibiotic masquerading or an antibiotic masquerading as a non-antibiotic. They seem to have a different mechanism of action. Interestingly, some of them do fall in between clusters. So you can see in the center, there's a group of these black dots that are in between the green cluster and the red cluster, meaning they share some mechanism of actions of these drugs, yet they seem to be very different than the standard antibiotic classes. So again, we had three alternatives. What we see is that for the most case, these are completely new mechanisms of action, or at least different enough from the standard antibiotic classes.
This is amazing because, again, the idea that we know how resistance happens to many types of antibiotics, and sometimes it's quite specific. ADP ribosylating kanamycin, for example, but that's not general. It doesn't happen to just any molecule. So when you have new antibiotics that aren't related to ones that are usable by the quiver of defenses that bacteria can develop, this is really exciting. And once again, the approach that you're using takes you away from, well, let's just modify streptomycin a little bit more. So this is wonderful. Wow.
Absolutely. It's very reassuring to see there are new and creative ways to kill bacteria. We don't need to keep on hammering on the same, killing mechanisms that the standard antibiotic classes use. Maybe there's a way in to kill them slightly differently. And hopefully, as you said, these new ways or these new molecules will not have resistance for them already in the clinical isolates because they kill bacteria very, very differently. Absolutely.
So are you aware of the Tiny Earth program?
No.
This is something for undergraduate students and sometimes high school, where they screen soil samples to look for bacterial isolates that appear to have antimicrobial activity. And as you might imagine, you have to figure out what's going on with those. And I'm not saying that your approach could be used directly, but I'm just thinking you could have this lyophilized sample. You have no idea how this works. And then you put it into your system and boom, I understand it's a lot of work to test them, but you would know what it was related to or unrelated to very quickly.
Mark, you're absolutely right. And that's exactly what we're trying to do, not using that program. We'll get to this, I hope, in the end. But we think we sort of unraveled here a way to quickly identify the mechanism of action and to find those promising leads that kill bacteria in a new and creative way. And we think this method can work not just with purified compounds, but with crude extracts, or even by co-culturing our library with a toxic bug that secretes something unknown into the media. And very early on in the drug development process, say, is this a promising direction? So we're all about exactly what you said now. Harnessing this approach for a very, very quick sort of decision, go, no go. Is this a promising lead? Is this, something that is not worth a while? It's another beta-lactam. And we want to do it with crude extracts or even with co-culture sort of conditions.
Let me take it still further. Sometimes these new antibiotics are actually not usable because they're super toxic to eukaryotic cells. And you would be able to know that this is something, forgive what I'm saying. I don't want to say, should you patent this, but this is a wonderful, wonderful, wonderful approach.
Thank you. Thank you. So I hope we'll get to the end of how we're trying. So this is just, hundreds of drugs. For what you're describing, we want to be able to pull off thousands of screens. And we think we can do it. We can leverage on AI, on neural networks, to achieve the capacity, the throughput to screen thousands of compounds and to quickly sort of classify them to interesting, not interesting, standard mechanism of action, dual mechanism of action, or a completely new mechanism of action. That is sort of the pie in the sky that we're aiming for. Absolutely.
I have students who isolate these things all the time, and we don't know how they work. There you go.
I'm looking constantly for people that we can collaborate with because we have this in place, it's running, and I would love to continue this discussion about
Thank you so much for saying so. This is amazing.
Thank you.
And I hope everyone understands. I mean, we've talked a couple of times in the podcast about the threat, the rising threat of antimicrobial resistance. And I threw some scary lines at you. We've had people that have talked about looking at possible AMRs that we'll find in different areas. So I hope all listeners and viewers see what an important concept that is, and the ability to quickly determine whether or not a new compound is similar to an old one or has a new approach is so critical, because many of the pharmacological companies don't see the money in well, they will now. They will now based on this, because otherwise it's very one at a time plugging things in. So I am very impressed.
Exactly. So I'll just, maybe I know our time is limited. I want to just show you, don't take it at face value. So I'm saying these are no mechanism of drug. Maybe not, who knows?
Sure.
The proof is in the pudding, right? So we said, let's follow up on a few of these non-antibiotic drugs and really show they target something else. So we can switch to slide number 9. Slide number 9 shows this network of drug-drug similarity exclusively for our non-antibiotic drugs. What emerges from that analysis is that it's not that each one of these drugs is its own snowflake and kills bacteria in a different way. They do cluster together. They have some common mechanisms of action. So you can see some do cluster here. Some do have connections within them. And we followed up on a few, and I'll just give you an anecdote so you don't take everything at face value. We followed up on a few of these drugs, these 3 drugs that you see here highlighted in red. They are what we call a network click. They connect to each other. They all seem to have the same mechanism of action, and we were lucky enough to be able to identify the binding partner of these drugs. They seem to all bind an initiation factor 2, that is related to translation, and initiation factor 2 is not currently a target of any of the standard antibiotics. So at least for one of these cases, we were able to resolve what is the drug target, and indeed it's a new mechanism of action, let's call it that way. Now, mind you, it's not a completely new way to kill bacteria. Mm-hmm. It interferes with translation, very similar to standard antibiotic classes, but it does it in a very different way than the tetracyclines and the macrolides and all these other antibiotics that target the 30S or the 50S. Okay, so let's, we have limited time. I want to switch to the future, right? So this is what we achieved now. Where do we see this going? And I think definitely people like you, Mark, can already sort of see where this is going if this can become a real high throughput. So if you switch to sort of slide number 10, just to give you an estimate, in the past, we were able to resolve the mechanism of action of a handful of drugs in an experiment using the cryocollection, for example. Now, with our approach, we can do it to 100 drugs, anywhere between 10 to 100 of drugs. That's what we can presently do with this pooled genetic screening that we do. We really want to push the envelope to another, order of magnitude to thousands of drugs, and we think that is actually needed. So again, I won't go into how exactly we're trying to do it. We're doing it right now, and it's working. We have initial observation that it is working, but I do want to sort of explain what is the motivation for being able to resolve this and why we think artificial intelligence is so critical for this sort of transition from the scale of dozens of drugs to hundreds and thousands of drugs. So the thought process there, and you can pull up slide 11, is under this pipeline of finding new antimicrobials using what we call phenotypic screens. So the thought there is that for decades now, we've developed new antibiotics or tried to develop new antibiotics by screening libraries of natural products, chemical libraries, bacteria, fungi that secrete different metabolites. And we really did what we call this phenotypic screening, where you were screening hundreds of thousands of molecules and seeing which ones of them kill bacteria and then trying to prioritize them. And that sort of process, which was very, very tedious, of finding which are the gems and which are just more beta-lactams, was extremely, extremely painful and caused that barrier, which we call the rediscovery barrier. What do we think we can do now to sort of circumvent that challenge? The idea there is you'll start off again with that phenotypic screen. You'll find those promising hits that kill bacteria, yet you still need to find a needle in a haystack, these molecules that kill bacteria in a new creative way. The idea there is that if we can increase the scale of our screening to thousands of different molecules, we can actually employ AI to do the classification for us. So the idea there is to use fully connected neural networks to do the drug classification and to train them on standard antibiotics. So we can take these profiles of sensitivity and resistance, plug them in, just like you plug a picture of a cat into a classifier, and that classifier tells you cat, dog, lion, or I don't know, right? So a very similar classifier that is based on a neural network will take that sensitivity and resistant profile of fingerprint as an input, and as an output will predict what is the drug class. Again, and will tell you, is it a standard mechanism of action? Is it a dual mechanism of action? Or is it a new mechanism of action? If we can do that using neural networks in a highly efficient way to do the genetic screens using robotics and next generation sequencing, we can really now take it to the next level, and in an order of weeks to months, cycle through thousands of different compounds and find those needles in the haystack, right? What are the promising leads out of all these hits that kill bacteria, yet most of them probably operate like standard antibiotics. So that is sort of the vision for where we see this working. And you were spot on, Mark. Let's try to work not just with compounds. Let's just take bacteria, co-culture them with our library, and see what metabolites they produce and how they kill our mutant library.
What an amazing program. I mean, I don't know if you can hear the applause, but it's there. If I put on my science fiction hat, you brought this up with neural networks and AI. My question is, can AI just go ahead and start designing you new antibiotics?
That's a great question. I think yes, and I think there are definitely groups already employing AI to design either new chemicals, new antimicrobial peptides that will have antimicrobial characteristics that will kill bacteria. I think there have been successes definitely using AI to design new molecules or new peptides that kill bacteria. Yet, I think there is a limitation there that these molecules tend to be more of the same, right? They're building on established molecules that kill bacteria in a certain way. Whether they can truly invent something completely new is to be seen. I think people in the field are definitely optimistic about it. And there are groups doing that, yet my own group does not sort of focus on that problem. We're trying to resolve how they kill and not to design completely new drugs using AI. But there are definitely some groups doing, I think, breakthrough work using AI to design new compounds.
I know that we're short on time, but this has been a remarkable session. I am so appreciative of hearing this. You know, part of why I do this podcast is to entertain myself. So, and today has been a wonderful example of that. And in addition, it gives hope in this battle to try and figure out what to do about multiply resistant organisms. Eventually, what are we going to do about XDR tuberculosis, which is resistant to almost everything we've got? Same thing with Neisseria gonorrhea. Now, you say, well, we're talking about E. coli, but then you're going to find new ways, hopefully, new approaches to try. That's very exciting.
Absolutely. And you can apply this approach with tuberculosis, right? You can build that library and work with transposon libraries, with Klebsiella. This sort of concept, this framework of how we approach mechanism of action can be reapplied to any pathogen or organism of choice. But as you said, antibiotics tend to work in the same way across organisms. The same drug works with a similar mechanism of action across a wide phylogeny, a wide sort of evolutionary distance. We think it will be the same mechanism of action in pathogens in our E. coli. Mark, I do want to do one last thing. I know
Please.
We're probably out of time. I do want to go to slide number 1 because I deeply believe, and I want to provoke some of your listeners to think about this slide. So if we can pull up slide number 1, it's not science anymore, right? It's an AI generated image. But I have to say, this is how I start most of my talks, and this is how I view the world. And I do want to share it with your audience. And again, maybe I'm an extremist, but we have to listen to extremists as well. My world of view is that we are at the cusp of another paradigm shift in biology. So just to describe the slide, what you can see is an AI-generated image of people celebrating on the deck of the Titanic. The Titanic is about to hit this iceberg looming in the distance, and I just carved there the initials AI, artificial intelligence. I think it really reflects this moment in time. I think we're about to hit this iceberg as a scientific community. It's not just, other professions that are about to hit this iceberg. It's us too. It's a major paradigm shift of how we do science, how we approach scientific problems. What we're trying to do with drug screening and mechanisms of action is just our attempt to apply AI to this sort of problem. But I think this is sort of an easy test case because it lends itself to neural network and for classification and standard approaches in computer science. I think if we meet again in 5-10 years, me and you, Mark, will realize that the way we do research in life sciences has changed to its core. And what I want to emphasize is we as scientists at all career stages need to start to think how we embed artificial intelligence into our research, how we keep on trying new things with artificial intelligence so we are prepared to be the scientists that we need to be 5 years from now. This is not looming, 25 years down the road. It's the next 5 to 10 years. We'll do that shift, and you can be part of that revolution or you can be left in behind. So again, I know I'm an extremist, but I truly believe that it's our role as sort of, the elder scientists in some communities to preach and to say, this is looming, we're about to hit this iceberg, you better prepare.
Well, that's a cautionary note, but it's an exciting one to a science fiction reader like me. So and in 5 years, it'll be our uploaded versions speaking to one another, right?
Yes, absolutely.
Yeah. Amir, thank you so much for a wonderful episode. You also have a wonderful photograph of your lab group. I love how you've worked really hard with so many people and you have all these broad interests. It's been such a pleasure chatting with you. And I want to wish you and yours the very best.
Thank you very much, Mark. I do want to just highlight that this work, I'm an ambassador. It was done by the students in my lab. through lots of hard teamwork. They are the people who deserve the credit. They did the work. They did the thinking. They did the analysis. They did the experiments. I was there for the ride, and it was a privilege to be there and to facilitate that journey. But it really was a team effort. This work was, primarily driven by Mariana Noto from my lab, but I have so many other extremely talented students. I can't mention all of them right now. You're very welcome to the lab webpage to see who are the people who did the real work.
And it's a wonderful webpage that we will link to in the show notes.
Thank you. Okay.
Thanks so much again.
Of course.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, especially this time, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Amir Mitchell can be found in the Program for Systems Biology at the University of Massachusetts Medical School in Worcester, Massachusetts. Many thanks, as always, to David Renata for superb expert editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.